Hello, everyone. My name is Chen, and I'm a third-year undergraduate here at UCR's Computer Engineering Department.
And I'm Helen Du. I'm a third year undergraduate student in the UCR Electrical Engineering Department.
Our project is titled Real-Time Object Tracking and Sorting of Particles for DNA Synthesis, and we're working under Dr. Philip Brisk from the Computer Science and Engineering Department, Dr. Victor Rogers, and Dr. William Grover from the Bioengineering Department.
So our overall project objective is to create a high throughput, low cost DNA synthesizer. To do this, we will be using a microfluidic manifold, which is basically just like a fluid channel, in which square microtransponders will flow through and be tracked and sorted in real time. The images below are traditional methods that have been used for DNA synthesis. So on the left, we can see a column-based solid phase synthesis method, which involves loading glass or silica beans into columns and delivering the reagents to the column to build the sequences. But this method involves many wash steps and thus creates a large potential of error due to the usage of only a single reservoir. On the right, we can see microwave technologies for large-scale DNA manufacturing. This involves spatially localizing the synthesis reservoirs and delivering the reagents to each of these reservoirs. However, this method also has substantial error rates, is very expensive, and does also not have the throughput necessary to meet the current demand for desired applications of synthesized DNA. Because of this, we will be using a different method to deliver the reagents. So instead of delivering it to reservoirs, we will actually be delivering the reagents to the solid base, in this case, the P-chip, which I will explain more further on. Now, Aaron will go into more detail about some of the applications that our project has.
So synthetic DNA can be used in synthetic biology for protein engineering for therapeutic proteins, as well as genetic network engineering. It can be also used as a medium for long-term data storage. It's a very high density media and is more stable than traditional media that's used for long-term data storage, such as tapes.
So I apologize for the laggy GIF, but the process of our DNA synthesizer works like the following. On the left, you can see a chip, this chip. This will act as a support for the DNA. These are actually square microtransponders called P-chips that were created by Pharmaseek, a company that we've been collaborating with for this project and was actually started by a UCR alumnus. So once scanned, we can see the P-chips start flowing to the right. It'll first get scanned by a purple light, which will allow the chip to emit a unique ID via radio frequency signals. We will then begin tracking the object to make sure that it's sorted into the correct reservoir for the desired nucleotide to be added, in this case, A, C, T, or G. The process will continue to loop until the maximum length of nucleotides is reached. In solid phase DNA synthesis, the maximum length is 200 nucleotides due to the chemical yield. Therefore, in order to create large sequences of DNA, we have to build many of the DNA sequences separately and assemble or ligate them together in the end.
Here you can see a picture of our current setup in the lab and the diagram of the simplified diagram on the left. The area labeled number 1 in the picture towards the bottom left is the syringe pump, and we use this in order to simulate the flow of fluid through the channel so that the P-chip can move left and right. Label 2 is a high-speed camera, which we're using for the acquisition of the image so that we can track. Label 3 and 4 are actually a close-up of the microfluidic channel setup. Number 3 is the P-chip itself, and 4 is the channel on which it flows. Label 5 is the backlight, which is used for the backlighting the channel in order to ensure a clear picture for our tracking. Label 6 is actually the FPGA module in which we do our image processing. Label 7 is the software interface that we use to control the whole system.
So currently, in order to track the P-chip, we are using a software called LabVIEW, which was invented by National Instruments to perform the real-time object detection and tracking on the P-chip. So to track the P-chip, we first need to retrieve the raw image from the high-speed camera that Aaron previously mentioned in his last slide. And next, we will perform image masking on the image in order to isolate the image so that there's only the P-chip. This is very important because it reduces the processing time since there are less pixels to search through in the image. Additionally, we will then threshold the image so that we can further isolate the image so that we can ignore all the pixels which are too bright or too dim. Finally, the actual detection of the P-chip is done by using a pattern matching algorithm, while the tracking of the P-chip is performed using a mean shift algorithm. Both of these algorithms work together to draw a bounding box around the chip as shown in image C, which provides us with the visual feedback we need to ensure that our program accurately detects and tracks the chip in real time.
Here we can see the data path for our image once it's processed. First, the camera sends the image to the host device. In this case, it's our computer. It's then sent through the acquisition logic and sent over to the FPGA to go through image processing that Helen mentioned in the last slide. Then it is sent back to the host device to go through object detection and tracking. Some advantages of running the image processing are that on the FPGA is that the FPGA can run tasks concurrently with the CPU, freeing up more processing power for object tracking and detection. The FPGA also solely focuses on image processing, as if you ran the image processing on the CPU, you may experience differing run times in between for each cycle, since it may be bogged down by other processes during context switching. We did a comparison study on running the image processing solely on the CPU and running the co-processing implementation on the FPGA, which we go into deeper detail on in the next slide. So as you can see here, the FPGA runtimes, which are the lighter gray bars on both of these graphs, the FPGA runs faster and more consistently with less variation between the runtimes than the CPU does. This is especially useful to ensure crucial frames aren't missed due to extended runtimes. A low variation is also crucial since it will allow us to more accurately predict that P-chips will be reliably sorted into their respective reservoirs without missing a couple of frames in between. A faster runtime also means that we can have a higher throughput, which means we can synthesize more DNA in less time. So following these results, we do all of our image processing on the FPGA, and we plan to move the detection and tracking algorithms also on the FPGA. We'd like to take a moment to recognize the other members of our team. Dr. Philip Brisk, our faculty mentor, our graduate researchers, Raymond Young and Jason Mandala, as well as Dr. Victor Rogers and Dr. William Grover. We'd also like to thank Pharmaseek, the company that we've been partnering with for this project, and who manufacture the P-chips that we use in this project. All right, are there any questions?
Thank you guys, excellent work. And feel free to stop recording if you like. I know you had asked before.